Question 1c asks us to discuss two ways that water temperature could affect the physiology of the fish in this experiment. Unlike warm-blooded animals, where the internal temperature of the organism is maintained at a constant temperature, the internal temperature of a cold-blooded animal comes to equilibrium with the external temperature of the environment. And this change in temperature relative to the environmental temperature can affect the physiology of a cold-blooded animal. Some things that we can expect to change physiologically is the metabolism. So the metabolism of an organism is the sum of all chemical reactions in the organism, such that when the temperature increases, the metabolism will also increase because of an increase in the kinetic energy and the movement of molecules throughout the organism. And at lower temperatures, when it's colder, a reverse effect will be seen, where a decrease in the diffusion rate and kinetic energy of the molecules will result in a decrease in the overall completion of the chemical reactions, and therefore a decrease in the metabolic rate of the organism. And why is that important? Because metabolism is linked to activity. The faster the metabolism, the more able the organism is to respond to its environment, and the quicker the organism can respond to its environment, and vice versa in lower temperatures. Another physiological effect we see in cold-blooded animals due to temperature is circulation of blood and also the heart rate. So there's an increase in the blood circulation and heart rate when the temperature increases, because of an increase in the kinetic energy, the movement of the molecules, as well as vasodilation. Vasodilation is the relaxing of blood vessels and arteries and capillaries in an organism. And the dilation occurs when the temperature increases, and vasoconstriction occurs when there is when the temperature drops, so when it gets colder. And this can affect both the heart rate and the blood circulation. The third thing I'd like to talk about is an increase in respiration when the temperature increases. And this is due to an increase in the ability for molecules to diffuse throughout the organism. So respiration is the exchange of carbon dioxide and oxygen between both the organism and its environment. And this occurs rapidly when the temperature increases and more slowly when the temperature decreases. And so here we can see that the temperature then will affect the respiration because of an increase or decrease in diffusion of gases. Question 2 is as follows. The flow of genetic information from DNA to protein in eukaryotic cells is called the central dogma of biology. Explain the role of the following in eukaryotic protein synthesis: RNA polymerase, spliceosomes and SNRs, codons, ribosomes, and tRNA. The central dogma of biology describes the flow of information from DNA to protein through RNA. DNA must be made into RNA because DNA cannot directly exit the nucleus into the cytoplasm. And the machinery to make the protein is located only in the cytoplasm. Therefore, mRNA is needed to move the message from the nucleus to the cytoplasm. The process of making RNA from DNA is called transcription, and the process of making protein from RNA is called translation. Specific cellular machinery is responsible for the synthesis of each of these molecules. So RNA polymerase catalyzes the conversion of DNA to RNA, and ribosomes located in the cytoplasm catalyze the conversion of RNA into protein, and that process is called translation. Now, when the DNA is catalyzed to RNA by RNA polymerase through the process called transcription, we get what is called pre-mRNA. This pre-mRNA has pieces of information within it called exons that do not code for protein. And these exons must be removed. The way that these are removed is through another set of cellular machinery in the nucleus called the spliceosomes and associated SNRMPs or SNRPs. So essentially, the spliceosome catalyzes the removal of exons so that we can ligate together the intronic sequences and get rid of the exons. This mature or messenger RNA then can move from the nucleus to the cytoplasm. Once it's in the cytoplasm, the ribosomes recognize the mRNA and start to translate protein. The tRNA or transfer RNA facilitates the movement of amino acids, which are the building blocks of proteins, to the ribosome so that these amino acids can then be incorporated into the protein. The tRNA recognizes specific codon sequences within the mRNA, and these codon sequences are units of 3 nucleotides for example, and the tRNA will then recognize these codons so that the amino acids can then be added to the growing protein polymer and ligated together. And this is catalyzed by the ribosome.